Welcome to Evolutionary Biology. Today, we are going to explore one of the most fascinating concepts in science, the RNA world hypothesis. We will journey back to the very dawn of life on Earth to understand how non-living chemicals first transitioned into the complex, highly organized living organisms we see today. This is the ultimate origin story. To understand why the RNA world is so important, we first need to look at a classic chicken and egg problem in modern biology. In our cells today, DNA acts as the instruction manual, holding all the genetic blueprints, while proteins are the robot workers that carry out physical tasks. The problem is, DNA cannot copy itself without those protein workers. But the protein workers cannot be built without the DNA instructions. So which came first? This paradox puzzled scientists for decades. This paradox brings us to the RNA world hypothesis, which provides a brilliant and elegant solution. Scientists propose that long before DNA and proteins existed, early life relied entirely on a single incredible molecule, RNA. RNA is structurally unique. It can store genetic information, just like DNA, but it can also fold into complex three-dimensional shapes to drive chemical reactions, acting as an enzyme. We call these RNA enzymes ribozymes. By doing both jobs, RNA bridges the gap between simple chemicals and cellular life. Now that we know what the RNA world is, let us look at why it matters today. This hypothesis is crucial because it explains the impossible simultaneous appearance of DNA and proteins. But it is not just ancient history. Right now, the ribosomes in your cells, which build all your proteins, are actually giant ribozymes. This proves that RNA still performs complex catalytic tasks. Scientists even use these ancient evolutionary principles to engineer synthetic ribozymes in the lab that can target and destroy viral diseases. Before we move on to the mathematics of RNA, let us visualize these dual functions. On the screen, you can see a diagram illustrating exactly why RNA is so special. On the left, the RNA molecule is folded into a complex shape, acting as a ribozyme to catalyze a chemical reaction, converting a substrate into a product. On the right, you see RNA acting as a genetic template, where its sequence of nucleotide letters is being copied. This single molecule is doing the heavy lifting of both DNA and proteins, solving our chicken and egg problem. Having seen how RNA functions, we must now consider the environment it lived in. Early Earth was incredibly harsh and chaotic. For these early RNA molecules to survive and evolve, they had to copy themselves. However, without modern proteins to help, this copying process was very messy and error-prone. Every mistake made during copying is a mutation. If an RNA molecule makes too many mutations, the essential blueprint to build a functional ribozyme is lost forever, halting evolution in its tracks. This challenge of copying accurately leads us to a fundamental probability formula. We can calculate the exact chance of an RNA molecule replicating without any errors. In this equation, P represents the probability of a perfect copy. The Greek letter mu represents the mutation rate, which is the chance of an error at any single nucleotide letter. Finally, L is the total length of the RNA sequence. Conceptually, 1 minus mu is the chance of getting a single letter right, and we multiply that by itself for every letter in the sequence. If the mutation rate or the length increases, the probability of a perfect copy drops dramatically. Now that we have our formula, let us see this with a concrete example. Imagine a primitive ocean containing a small, basic ribozyme that is exactly 50 nucleotides long. The chaotic environment causes a copying error at a rate of 0.01 per nucleotide. We want to find the probability of a perfect copy. We substitute our known values into the formula. The chance of getting one nucleotide correct is 1 minus 0.01, which is 0.99. We then raise this to the power of 50. The final answer is approximately 0.605. This means there is a 60.5% chance the ribozyme will produce a perfect, fully functional clone of itself. So, we have seen that a short RNA molecule has a decent chance of surviving. This brings us to an important evolutionary hurdle. As RNA molecules evolved to perform more complex tasks, they naturally needed to become longer to store more information. But remember, early replication was sloppy. Because the sequence is longer, there are many more opportunities for a fatal error to occur during copying. This tension between needing to grow and the risk of mutations introduces a critical concept known as the error threshold. Let us apply our formula again to understand this error threshold. Suppose our new, highly complex ribozyme requires a length of 200 nucleotides to function properly. The chemical environment has not changed, so the mutation rate remains at 0.01. We plug our new length of 200 into the equation. The chance of a single nucleotide being copied correctly is still 0.99, but now we raise it to the power of 200. The overall probability drops much faster than one might intuitively expect, giving a final answer of approximately 0.134, or 13.4%. This physically means most copies will fail. This drastically low number we just calculated reveals a massive evolutionary roadblock. With only a 13.4% chance of success, nearly 87% of all offspring will be defective mutants. The error threshold is the maximum size a genome can reach before mutations destroy information faster than it can be copied. Without better copying mechanisms, RNA molecules simply could not grow long enough to store the vast amounts of information needed for complex life. This mathematical limitation is exactly why life eventually had to transition from the RNA world to the more stable DNA and protein world we see today. To wrap up our journey, let us review the most important ideas. First, the RNA world hypothesis elegantly solves the origin of life paradox, showing that early RNA acted as both the genetic blueprint and the chemical catalyst. Second, we learned that replication fidelity is a delicate balance between mutation rate and sequence length. Finally, the high mutation rates of early Earth created a strict error threshold, limiting how long RNA could grow. This very limitation is what ultimately drove the evolution of the complex, interconnected DNA and protein-based life that covers our planet today. You now have a solid understanding of the dawn of life.